So I'm going to be going over my DNA model for RLA5. This is what it looks like. I made it out of hockey tape, just white, black, and clear tape. I'm just going to be going over what it does well and what it does not do well, just as we did in class with the several different models. First, going over what it does well. It shows the deoxyribose and the phosphate backbone, as you see on the sides here. The deoxyribose is the brown segments, and the phosphates are the white segments in between. The deoxyribose are seen in this spot because it holds together the bonds, the AT and the GC base pairs. Another thing it does well is shows the hydrogen bonds. The hydrogen bonds are what holds together the base pairs, the ATs and the GCs. That's being seen here with the clear tape between each of the bonds as you go down the ladder. It shows anti-parallel. As you see here, I have a 5' and a 3' end, and then a 3' and a 5' end. Anti-parallel just means that they run in opposite directions, and since I labeled that here with the 5' and 3' ends, it shows that it is anti-parallel, and then it has 5' and 3' ends. And then it also shows the nucleotides, as it's just this whole segment here that includes the phosphates, the deoxyribose holding together the base pairs along with the hydrogen bond. It shows the nucleotide. And then what it does not do well, or does poorly, show a double helix. I mean, if I bent it and kind of twisted it, it still kind of just doesn't really do that very well. So it doesn't really show the double helix shape. It doesn't really show the double helix shape as well as other models do. Even when I bend it, still doesn't really show it. And along with that, doesn't show minor and major grooves, which would be, if it was in the double helix shape, the spacing between each of the twists in the shape. But my model doesn't really show that very well. It does not show the AT and GC base pairs, which are the purines and pyrimidines. You see, it shows that it has base pairs, but it doesn't actually label which one's AT and which one's GC. If I had different color tape, for example, say I had a red and a blue, and then a green and a yellow, the red and blue could be A and T, and the green and yellow could be G and C. And that would show complementary, that the A and T's do go together and the G's and C's do go together, but my model does not show that. My model also doesn't show the phosphodiester bonds, which would be the actual bonds that hold the backbone together. I mean, I guess you could argue that the lines here could be it, but I don't think it shows it very well, or easily. I guess you can't really pick it out, but yeah, that's basically it from the list that we need to show in our diagram. I think it does show more poorly rather than does well, but maybe if I had different resources, such as more colored tape, I just didn't really have that. I could make the base pairs, each color represents an A and a T, a G and a C. But yeah, this is my model.